Plant biotechnology and its method. Plant biotechnology is a powerful tool for the development of new plant traits and varieties. Such new varieties must be produced on a large scale to achieve commercial success and to satisfy the demand from growers. Traditionally, new varieties were achieved by the seed propagation methods. Currently, micropropagation produced plantlets offer a practical alternative for many plant species. Figure 1 shows plant in vitro propagation that is available in the link from the Springer. In this photo, you can see from the cactus, it's the organo, organogenesis and collagenesis. And after the organogenesis, we have a micropropagation and also we have a hairy root culture from two different ways. From the micropropagation, we have an open field and we have a vertical garden and we have a hairy root culture. Also, we achieve bioactive compounds. From the collagenesis, we have a cell suspension culture, and from this we have elicitors, persergers, abiotic esterase, and then we achieve bioactive compounds. Also, we can do genetic modifications by basics, by Argobacterium tumefaciens, and also by gene editings. Micropropagation is labor intensive, although the time needed to commercialize new development in genetic engineering and molecular biology techniques allowed um, uh, excuse me, micropropagation is labor intensive, although the time needed to commercialize new varieties and permit the production of disease-free plants is reduced. Recent development in genetic engineering and molecular biology techniques allowed the production of improved and new agricultural products in several countries worldwide. This is only possible with the evolution of tissue culture techniques, which provide the platform for the introduction of genetic information into plant cells. Current plant biotechnology has developed as a new age of science and technology where the production of secondary metabolites, valuable plant genetic improvements, germ germplasm's conservation and production of large number of disease-free and new varieties are preferred. Production of artificial seeds, biopharmaceuticals, plant-made plants, starting with antibiotic-resistant tobacco in 1982 and plant-made vaccines or antibody plantibodies is part of the current research work in plant tissue culture science. Most of the plant tissue culture industries are exploiting the fundamental property of regeneration of plant cells for rapid production of elite varieties with superior genotypes on a large scale in a comparatively short time. Initial problem: search for an isolated cell model, validation of plant cell totipotency, and the current plant tissue culture industry is estimated to be worth around $150 US billion to 50 to 60% of the agricultural industry. Annual demand of tissue culture raised products is about 10% of the total estimated value. Most of the plant tissue culture based industries are limiting the time consuming labor or manual processes fully by transforming into more automated and robust technologies. Agrobacterium tumefaciens and the related Agrobacterium species have been known as plant pathogens since the beginning of the 20th century. However, only in the past 2 decades has the ability of Agrobacterium to transfer DNA to plant cells been harm seeds to be harm to be harnessed for the purposes of plant genetic engineering. Since the initial reports in the early 1980s using agrobacterium to generate transgenic plants, scientists have attempted to improve this natural genetic engineer for biotechnology purposes. Some of these modification species, uh, excuse me, some of these modification has resulted in extending the host range of bacterium to economically important crop species. However, in most instances, major improvement involved. I think if I rotate it, it will be better. Uh-huh. It's better a little bit. Okay, major improvement involve alteration in plant tissue culture transformation. However, in most instances, major improvement involved alteration in plant tissue culture transformation and regeneration condition rather than manipulation of bacterial or host genes. Agrobacterium mediated plant transformation is a highly complex and evolved process involving genetic determinants of both the bacterium and the host plant cells. Knowledge of fundamental biological principles embracing both the host and the pathogens have been and will continue to be key to extending the utility of agrobacterium for genetic engineering purposes. The genus Agrobacterium has been divided into a number of species. However, this division has reflected for the most part disease symptomatology and the host range. Thus, a radiobacter is an avirulent species. A tumefaciens causes crown gall disease, a rhizogen causes hairy root disease, and Agrobacterium rubi causes cane gall disease. Okay, again, I review it. The genus Agrobacterium has divided into a number of species. However, this division has reflected for most part disease symptomatology and host range. Thus, Agrobacterium radiobacter is an avirulent. Agrobacterium tumefaciens cause crown gall disease. Agrobacterium rhizogen cause hairy root disease, and Agrobacterium rubi cause a cane gall disease. Ruby cane gall disease. More recently, a new species has been proposed, Agrobacterium vitis, which cause gall on a vitis grape, and a few other plant species. Although Bergey's Manual of Systematic Bacteriology still reflects this nomenclature classification is complex and confusing. We know that symptoms follow for the most part. The type of tumorogenic plasmid contained within a particular strain, curing a particular plasmid and replacing this plasmid with another type of tumorogenic plasmid can alter disease symptoms. For example, infection of uh, plant with Agrobacterium tumefaciens C58 containing the nopalin type T plasmid PTIC58 result in the formation of a crown gall teratome. When this plasmid is cured, the strain becomes non-pathogenic. Introduction of RI or Re plasmid into a cured strain converted the bacterium into rhizogenic strain. Furthermore, one can introduce a T plasmid or tumor inducing plasmid from Agrobacterium tumefaciens into a Agrobacterium rhizogenic. The resulting strain incises tumors of altered morphology on Kalanko plants. Introduced can One can introduce a T plasmid into rhizogenic resulting strain, altered morphology of Kalanka plants. Okay, now we have one figure that's a production of a transgenic plants. That's also available in these links. How we can understand it. This is a plant. If we have Argobacterium tumefaciens here. Okay, this bacterium this plasmid has a two region, T plasmid and tDNA, virulent and tDNA, bacterium genome. tDNA, now this is a symptomatic. Kerongal is shown here. Continued T plasmid, it's written here. Contegrate T plasmid on the plant cell. Transformed cell. Kanamycin to be antibiotic resistance, to determine how many bacteria or how many plasmid is transformed. Now we have a cultured cell plant, and it's a transgenic plant. Now the cell of a transgenic plant. And what is written here? It's a foreign DNA, kanamycin, and integrated site. If we have a plant and we show that this and we have a cane, now if we have a kanamycin antibiotic resistance, okay, it's very difficult to understand it. Actually, we want to prevent the Krongal. We want to prevent the Krongal. We need the transgenic plants. How to produce transgenic plants? We add one fragment or foreigner part of the DNA here. It's very hard for me to understand. The molecular basis of genetic transformation of plant cell by Argobacterium is transfer from the bacterium and integration into a plant nuclear genome of a region of a large tumor-inducing T or rhizogenic Re plasmid resident in Argobacterium. As discussed in more detail below, T plasmid carrying strains of Argobacteria induced not only plant tumor formation but also production of various amino acid and sugar phosphate derivatives termed opins. The transformed plant cell secretes opins, which can then be taken up and catabolized for use as a food source by the infecting bacterium. The T plasmid carries the genes for open synthesis by plant cells as well as the corresponding catabolism genes. T plasmids traditionally have been classified by open type, and here we will retain this classification scheme with a focus mainly by the two best characterized T plasmid designated as the octopin and nopalin type. Several octopin type PTA6, B6, AC5, AC5, 955 and R10 and nopalin type PTC56, 58 and PT37. T plasmids have been expensively characterized and many have been sequenced at this time. All T plasmids code for functions associated with I or induced plasmid replication and the reception of exogenous signals released by the plant host and neighboring agrobacterial cells at the site of infection. Genes encoding each of these functions are generally clustered on the T plasmid with the exception of two spatially distinct regions, the virulence or vir region and the transfer DNA or tDNA required for infection of plants, and the TRA and TRB regions required for conjugative plasmid transfer. The following sections summarize our current understanding of the T plasmid encoded function. Okay, in the schematic of octopin type, T plasmid PTIA6 is shown on figure 3. In this T plasmid, we have a one virulent region. In the virulent region, we have a sensor kinase virulent A, translocation virulent B. We have a tDNA processing and a recurrent virulent C, virulent activator virulent G. We have a tDNA processing and transport virulent D. We have a tDNA movement virulent D. We have a tcDNA and we have a tRDNA, LB, On the plant, we have auxins, cytokines, and opins. Opins, yes. We have a TRA, T plasmid processing. We have a opin catabolism, opin uptake and degradation. We have a TRB, T plasmid conjugation. And for the for replication, we have a T plasmid replication. Schematic of octopin type T plasmid or tumor induced plasmid PTIA6 showing location of genes coding for plasmid maintenance replication. Infection of plant cells, virulent region, tDNA cell survival in the tumor environment, open catabolism, and conjugative transfer of the T plasmid to recipient agrobacteria TRA and TRB. The various contributions of the virulence gene produced to tDNA transfer are listed. TL, tLDNA, and tRDNA are delimited by the ORI-T-like border sequences, black boxes, RB right border, LB left border. OD overdrive sequence, white boxes, enhances virulent D2 relaxes nicking at the tDNA border sequences when derived to plant cell and integrated into the plant nuclear genome. tDNA code for biosynthesis of auxin and cytokine resulting in proliferation of plant tissue and production of opins that serve as nutrients for the infecting bacterium adopted from reflection. Okay. In the overview of the protoplast isolation technique, healthy 5-week old plants, thin slicing of leaves with sharp razor, filtered enzyme solution, and then incubating cut leaves in enzyme solution, protoblast in round bottom tubes, and then cut tips used for handling protoplast. During the last decade, a very extensive technology on plant protoplast has been developed. The introduction in the 1960s of methods for isolation of viable protoplast by enzyme treatment made it possible to obtain sufficient quantities for experimental purposes. Protoplasts are usually isolated by treating tissue with a mixture of degrading enzymes in solution, which contain osmotic stabilizer to preserve the structure and viability of the protoplast. The relative ease with which protoplast isolation can be achieved depends upon a variety of factors. The most important of these are the physiological state of the tissues and cell materials, the choice of enzyme, the composition of the solution, and the concentration and type of osmotic stabilizer. After isolation and protoplasts are suspended in nutrient media with adjusted osmolarity to maintain viability and structural integrity. The culture solution contains mineral nutrients, vitamins, carbon source, and growth hormone, as well as osmotic stabilizer. For survival, the protoplast also require organic nutrition sources and possibly coconut milk and various metabolites. Protoplasts are usually cultured in liquid media in droplet unit cell clusters have formed, after which they are plated on nutrient agar. Protoplasts have been employed extensively in studies on plant virus uptake. Much of the research has been performed with leaf protoplast of um, N. tabacum. New insights have been gained on the uptake infection process. One end of the virus attaches itself onto the membrane, TM. The uptake then appears to processed by an endocytosis on the detail of the process. In a population of protoplasts, up to 90% become infected and the replication occurs nearly exponentially after a delay of 6 to 8 hours. The protoplast systems have also enabled investigation on virus species specificity and strain interaction, as well as aspects of molecular biology which was not feasible with plant tissue cells. Recently, another approach has been followed to incorporate foreign nucleic acid. Recently, another approach has been followed to incorporate foreign nucleic acids into plant protoplasts. Matthew et al. Reported using lipid vesicles, liposomes, to sequester bacterial RNA to incorporate the RNA into carrot protoplasts. Incubation medium which had been treated with the sequesters 3H contain a large amount of 16S and 23S RNA which was still intact. However, uh, RNA extracted from the supernatant fluid of a protoplast which had been treated with the liposome sequestered RNA contained some intact RNA. The authors concluded that the RNA breakdown which occurred after lysosome mediated uptake into the protoplasm might have taken place because uptake occurred through endocytosis of the intact liposomes rather than fusion. This could result in the liposomes being rapidly degraded by the lysosomal apparatus. When uptake occurs through fusion, the interaped molecules are extruded into the cytoplasm, thus escaping degradation by the lysosomes, although the liposome sequestered RNA is not totally protected inside the plant protoplasts. This approach does seem to present a gene transfer method worthy of further study. Conventional crop breeding has thus far been sufficient to avert the dire Malthusian prediction of food shortage for a growing human population. Most projections suggest that novel yield enhancing solutions are needed to avoid global crop production reaching a plateau. Genetic manipulation has been used to successfully engineer simple traits such as insect and weed resistance. More refined molecular tinkering holds the promise of spectacular gains if fundamental pathways are targeted. Photosynthesis is one such pathway. Okay, environmental factors such as light, carbon dioxide, temperature, wind, water, and nutrient, and engineering photosynthesis like optimizing Rubisco, optimizing Calvin-Benson cycle, introducing of a carbon concentration mechanism CCMS, introducing pyrenoid-based CCM, introducing cyanobacterium carbon dioxide bicarbonate pumps. On the chloroplast, we have oxygen and carbon dioxide to Rubisco. We have a pyrenoid CCM. We have a Calvin Benson cycle. We have a nucleus. We have a photo respiration, peroxisome, mitochondrion, carbon dioxide, bicarbonate, and we have an increased growth and yields. And this is a current crop. In the current crop, it's low, and the increased growth and yields we have it if we do this. Number of rows is being studied to improve photosynthesis in crop plants. These include improving in the efficiency of the primary carbon dioxide fixing enzyme Rubisco, optimizing elements of the Calvin Saguri Calvin Benson cycle, introducing the carboxysome based carbon concentration mechanism from cyanobacter, introducing an algal pyrenoid CCM and improving the photochemical response of photosynthesis to rapid change in light condition. Further studies are also related to attempt to convert C3 crops such as rice to the more efficiency for type photosynthesis. Synthetic biology is making large steps in engineering alternate CCM mechanism into higher plants. In addition to effort in manipulating elements of the Calvin-Benson cycle, already present, although mesophyll conductance to carbon dioxide had been a relatively overlooked limiting factor until recently. It is now considered a promising potential target for increasing photosynthesis. It seems clear that expanded research efforts are currently required to build upon many of the technologies described above by, for example, enhancing our understanding of CAM metabolism and introducing alternative non-plant CCM into C3 plants. Overcoming these challenges will require sustained investments in long-term research programs, with some of these research areas currently being advanced through large-scale privately funded projects. Ultimately, in the case of C4 rise, necessity for development in a C4 species as well as sufficiently for engineering C4 rice will need to be considered when determining gene function. Existing candidate regulators are currently being functionally validated, and this is ongoing. Knockdown experiments in maize and cereal are examining necessity, while overexpression in rice is being used to scrutinize sufficiency. Future advances in engineering C4 rice will need to involve integrated analysis of these experiments together with further comprehension of the related gene regulatory networks. We posit that the we posit that the successful integration of these different characteristics as discussed above, coupled with the development of a strategy of how the three C3 plants could be genetically altered genetically altered, allowing both the introduction and the establishment of the C4 pathway should be a significant breakthrough in the field of synthetic biology. Recent advances and ongoing incremental findings suggest that the improved crop photosynthesis could assist towards feeding a growing population in the near future. Plants with favorable characteristics have been produced for thousands of years by conventional breeding methods. For example, inbreeding for production of killing lines, desirable traits are selected, combined, and propagated by repeated sexual crossing over numerous generations. This is a long process taking up to 15 years to produce new varieties. Genetic engineering not only allows this process to be dramatically accelerated in a highly targeted manner by introducing a small number of genes, it can also overcoming the barriers of sexual incompatibility between plant species and vastly increase the size of the available gene pool. Transgenic GM plants are those that have been genetically modified using recombinant DNA technology. This may be to express a gene that is not native to the plant or to modify endogenous gene. The protein encoded by the gene will confer a particular trait or characteristic to that plant. The technology can be utilized in a number of ways. For example, to engineer resistance to abiotic stresses such as drought, extreme temperature, or salinity, and abiotic stresses such as insect and pathogens that would normally prove that normally prove detrimental to plant growth or survival. The technology can also be used in the developing world. New generation GM crops are now also being developed for the production of recombinant medicines and industrial products such as monoclonal antibodies, vaccines, plastics, and biofuels. In 2007, for the 12th consecutive year, the global area of biotech crop planted continued to increase with a growth rate of 12% across 23 countries. The principal crops grown are soybean and maize, although cotton, canola, and rice are also on the increase. However, genetically modified crop grown in the EU amount to only a few thousand hectares, 0.03% of the world production. 6, which is probably a reflection of the European opposition to this technology. In contrast, food derived from the GM plants is uh, ubiquitous in the USA. Indeed, many animal feed used in Europe derived from imported plant material contain GM products. Similarly, GM cotton is widely used in clothing and other products. In the developed world, the nutritional content of food items is not of major concern, and as individuals have access to wide variety of foods that will meet all of their nutritional needs in the developing world. However, this is often not the case, with people often relying on a single staple food crop for their energy intake. GM technology offers a way to alleviate some of these problems by engineering plants to express additional products that can combat malnutrition. An important example of the potential of this technology is the Golden Rice Project. Vitamin A deficiency is widespread in the developing world and is estimated to account for the deaths of approximately 2 million children per year. In surviving children, it has been identified as the leading cause of blindness. Humans can synthesize vitamin A from its precursor beta carotene. Humans can synthesize vitamin A from its precursor beta carotene, which is commonly found in many plants but not in cereal grains. The strategy of the Golden Rice Project was to introduce the correct metabolic steps into rice endosperm to allow beta carotene synthesis. In 2000s, Ye et al. Ye et al. Engineered rice that contains moderate levels of beta carotene, and since then research have produced the much higher yielding golden rice too. It is estimated that 72 grams of dry golden rice too will provide 50% of the RDA, or recommended daily allowance of the vitamin A, for a 1 to 3 years old child. Initially, golden rice was developed for farmers in the poorest countries, and from the beginning, the aim of the scientists was to provide the technology free of charge, which required the negotiation of more than 100 intellectual and technical property licenses. Okay, and that is a reference of this article. Okay, I learned a lot. Now I should back to my exam. Can I answer this question? Which of the following can be used to obtain callus? To introduce correct metabolic steps into rice? Does to allow to introduce correct metabolic steps. Okay, bye bye.